Hello everyone and welcome back to Null for GCSE. In this video we're going to go over B6 and also just a note that you can actually access the document which has all of the questions on my website. So there's a link on my website and for free you can download them and answer them with me as you go through the video. But let's get started. So this is quite a big topic. This video will be split into timestamps below. So firstly, compare the advantages and disadvantages of sexual and asexual reproduction. Let's begin. So I would recommend pausing the video and having a go before looking at the answers. So sexual reproduction advantages include there being genetic variation. The species can therefore adapt and the chance that they'll get a disease decreases because it's less likely to affect a population. Disadvantages include that it requires time and energy in order to find a mate, and it's not possible if you are isolated. Asexual advantages is that it allows a very rapid population increase, only requires a single parent, is very time efficient, so overall it's quite much faster. Disadvantages is that it may only be suited to a particular habitat, so there might not be any genetic variation and therefore a disease could affect the entire of the species. The next question, explain sexual reproduction in flowers. Now I've included this question because most people don't tend to revise it for the exam. So this is a maximum that you're probably going to be expected to know. So you just need to know that the flowers make both the male, the pollen, and the female, the ovule gametes via meiosis. The pollen of one flower then pollinates the ovule of another flower, and this then forms seeds which grow into new plants. So make sure you're aware of that. The next subtopic, meiosis. Describe three differences between the processes of mitosis and meiosis. So mitosis produces 2 daughter cells, however meiosis produces 4 daughter cells. So there's only one cell division that occurs in mitosis, but there's actually 2 cell divisions that occur in meiosis. Mitosis produces cells with 2 of each chromosome, so the mother and the father. But meiosis is producing cells with only 1 of each chromosome, so they're producing the gametes. Mitosis produces genetically identical cells, and meiosis is producing genetically different cells, individual cells. And the final question about this topic, describe one similarity between the 2 of them, and you could say that the DNA is doubling or being copied or replicated once, or you could say that there's an increase in the number of mitochondria, increase in the number of ribosomes, or any other subcellular structures that follow that pattern. The next subtopic about the DNA and the genome. Explain how a change in one amino acid in an enzyme molecule could stop the enzyme from working. So you need to know that enzymes, in terms of amino acids associated with proteins, so one amino acid change would cause different proteins to be made because proteins are chains of amino acid. This would cause the active site to change, and therefore the substrate would not be able to fit, and therefore it would not bind to the active site. It wouldn't break down the proteins, and therefore it wouldn't work. The enzyme wouldn't work. The next subtopic, DNA structure and protein synthesis. So firstly, define variation, and then look at what process causes variation. So link to your knowledge. So variation is the differences between organisms, and it's caused by the early stages of meiosis, where you've got the homologous chromosomes that are exchanging their DNA, and this is called crossing over, and that causes variation. Now you do not need to know any further than this, so if you are unaware of this, then just make sure you memorize these 2 sentences here. What are homologous pairs? Well, homo always means same, so it means that they're matching pairs. So there's the paternal and the maternal pair. So for example, if you have a male I gene and a female I gene, now they're the same pair, so they're the matching, but it's just from paternal and maternal. So both chromosomes are coding for the same gene, but they're just different versions, so they're kind of alleles. The next question, what are base pairs connected by? And that answer would be hydrogen strands. What are the non-coding regions? Because you often hear coding and non-coding. So non-coding regions are the ones that are actually switching the genes on and off to control what the coding regions are able to produce. So the coding regions are actually telling you what gene you're going to have. For example, it could be the blue-eye gene or the black-eye gene or the brown-eye gene. But the non-coding region is saying that you could have the blue-eye gene, but it switches it off. So that's its function. The next question, why is DNA defined as a polymer? So this requires you to talk about your knowledge of what a polymer is, and quite simply you can just say it contains many nucleotides, and these nucleotides act as monomers, and multiple monomers bonded together act as a polymer. The next question, which is a 4-marker, describe the structure of a nucleotide. So the structure of a nucleotide goes as following. You have the phosphate, which is attached to the sugar. This has one of 4 bases, and the bases are A, C, G, and T. Moving on to the next subtopic, define what mutation means. So mutation is defined as an error in the sequence of DNA that usually occurs during cell division. This can then cause a new allele to be formed. Okay, so an error happens in the DNA sequence. This occurs during cell division and causes a new allele to be formed. Not all mutations are bad, but they have the potential to be if they're unable to perform their function. Evolution. Now we've got a 6-marker. The eyes of some birds contain cells that detect UV light. UV light is detected by some fruits and the urine of some animals. Explain how birds that can detect UV light have evolved from birds that could not detect UV light. So it's asking you to explain the process of evolution and then apply this to this specific case of birds. So, quite a hefty answer here. You've got the mutation occurring within the gene or DNA, which randomly occurs due to chance. This then causes a new or different protein, for example, a visual pigment to be made in the retina of the bird. You've then got the variation in the wavelengths of light that birds can see. The birds with the mutation, or the ones that are able to detect the UV light, are more likely to see the fruit. And the birds with the mutation, that's just repeated there. And therefore, they get more food. So some mammals or fruit, and they actually avoid being eaten by small mammals because they can see their urine. So they are out-competing the birds that do not have the mutation. So this mutation has benefited those that have received it by chance. And therefore they are more likely to survive and reproduce and have offspring by natural selection. So hopefully you can see you've got mutation, which leads to the physical change, which leads to either being a benefit or a detriment, and a survival advantage, which leads to natural selection. The next question. Describe how the ancestors of modern lemurs have evolved into the species shown. So obviously you can't see the picture here, but it's asking again the process of evolution. Oh, and another thing, always remember to say that natural selection occurs over many generations until the gene is fully developed. So in terms of this evolution, it has occurred because of the isolation of different populations, and then there being variation within the 2 habitats and within the populations, and the genetic variation or mutations happen, which mean that those that have the mutations are better adapted to survive. They pass on those favourable allele characteristics, and eventually they cannot produce fertile offspring with other populations, and that is when a whole new species has been formed. The next subtopic, selective breeding. Describe the steps to selective breeding. So firstly, you've got the desired characteristic which is found. The organisms are then bred together, and only the offspring that exhibit the desired characteristics are then bred again. This is repeated and repeated over several generations until the desired trait is fully developed. Evaluate the advantages and disadvantages of selective breeding. The advantages include there being an extra economic gain. For example, a cow that produces more milk, that's economically useful to the farmer. It could eliminate diseases. They could try and reproduce animals which have a certain mutation against a disease. It creates new varieties of crops, of course. It doesn't require genetic modification. It doesn't require any science. It's just selectively breeding specific species. And it produces stronger animals. Disadvantages, however, animal discomfort if you're forcing them to breed together, the lack of variety and variation within them. If they're all going to have the same genes, that could make them really susceptible to diseases as well. There's no control over a genetic mutation, and it could lead to inbreeding, which, if you ever heard of the mad cow disease, that was caused by that. Okay, the next one. Name 2 qualities that pet owners may want. Firstly, docile, which just means they're very calm, they're not very loud and boisterous, or that they're aesthetically appealing. The next subtopic, genetic engineering. What is the purpose of genetic engineering? So, genetic engineering is modifying the genome of an organism by introducing a gene from another organism in order to give that organism the desired characteristic from another. For example, plants have been engineered for disease resistance, or bacteria has been engineered to produce substances that are useful to humans. For example, bacteria have been engineered to produce insulin, and this can be given to diabetic patients. The next question, explain the process of genetic engineering. So this is obviously very well and thoroughly explained, but because it's a full marker, you don't have to put everything, but I would recommend putting as many as you can because you can't get demarked if you put something wrong. So the genes from the chromosome are cut out using restriction enzymes. This then leaves what we call sticky ends, unexposed pairs. The virus or bacterial plasmid is then also cut using that same restriction enzyme to create sticky ends. And this also contains an antibiotic marker gene. This is a thing that you don't really need to put. The loop and the gene both contain sticky ends, and they are then now joined together by another enzyme, a ligase enzyme. And now you have this loop, which contains a bit of a virus and a bit of a chromosome. The combined loop is then placed inside of a bacterial cell, and it then multiplies within that, and it will now contain the modified gene. So as the bacteria grows, you can then see which ones are resistant to an antibiotic, which ones aren't, and the ones which are, you can see, have been genetically engineered. And you can see this start to, the colonies start to group together, and the ones that have produced the modified gene would obviously survive longer. And in terms of plants, so within plants, if we're talking about genetic engineering, the vector is then put in meristematic cells, so specialized cells, which can then produce identical copies of that modified plant. Moving on to the next subtopic, cloning. So describe the process of tissue culture, which is also known as micropropagation. So just be aware of that. I hope you can see how we're going through all of the different things. So selective breeding, genetic engineering, tissue culture. These are the key things that you need to be able to know. So tissue culture is when there's a small tissue sample that is taken from the parent plant. The tissue is then grown in an agar plate or in a nutrient broth, and the plant hormones then begin to form a big mass of tissue. So you've got the small cells grow into a clump of cells, and you've then got different mixtures of hormones and nutrients that you can put, and it will then result in identical plantlets. And once the roots have grown, they're then transferred to a greenhouse or to the soil to grow. The next one is taking plant cuttings, which is a little bit similar to tissue culture. So it's when you cut the stem from a parent plant, you then dip the stem in rooting powder, which contains loads of hormones that we'll look at later, and you then plant it, and the plant then grows as a clone, However, an issue with taking plant cuttings is that it is a slow process. You cannot produce many at once because you don't want to kill the original plant either. Okay, describe the process of adult cell cloning. So the nucleus is removed from the unfertilized egg, and the nucleus is then taken from an adult body cell. So you've got a cell, an unfertilized cell, you're taking the nucleus out, and you're then inserting a nucleus from the adult body cell, the thing that you want cloned. You then give it a tiny zap of electricity, which stimulates it to start dividing into an embryo, a clump of cells. This then contains the exact same genetic information as the adult body cell. And when developed, you then can insert it inside an adult female womb in order for it to grow, develop, and then later the woman give birth. And that's how you can get a clone. So if you've ever heard of Dolly the sheep, that's exactly what happened. And it worked. Describe the process of embryo transplanting. So in terms of the context of cows, so the best cow would be given fertility drugs to increase the number of eggs. Sperm is taken from the best bull and used to fertilize those eggs. Embryos are then separated into individual cells which can then go on to form new embryos. The embryos are then implanted into the surrogate cows, and those cows then give birth to clones. So what's the advantages and disadvantages? Because these two are both types of cloning, so that's why it's in the same box. Advantages include that for difficult species to grow, it can actually be started. It can be planted at any time of the year in order to sustain those endangered species, and you can use it to produce large numbers of populace. Disadvantages is that there's a lack of genetic variation. It's extremely true because they're identical, and it changes the stable environment. There could be deadly consequences that we don't know yet, and animal discomfort. Moving on to the next topic, speciation and the history of genetics. So firstly, you need to know what Mendel discovered. So Mendel discovered that some characteristics are controlled by a single gene, and each gene might have different forms, called allele. So alleles are different versions of the same gene. And the alleles can be in one of two states, dominant or recessive. And then you have Watson and Crick, and they both discovered the double helix structure of DNA. Evidence for fossils and evolution. So information about extinct animals is often not clear because the fossil record is incomplete. Give 3 reasons why the fossil record is not clear for older species. Okay, so the first thing you could put is that the fossils have been buried deep, so they're very hard to find. The fossils are very small, so they're hard to find. They're more likely to have been destroyed by geological activity, earthquakes, erosion. The oldest organisms were also very soft-bodied, so most of the tissue would have decayed by now. And dating older fossils is actually very hard. Older eras were less researched by scientists, and as we're moving more and more into the future, it's getting harder and harder to find information about the past. Also, it's unclear when one species evolves into another because, there's not enough fossils to highlight this. So that's why evidence of evolution is difficult to find. The next question. A mass extinction is a rapid decrease in biodiversity on Earth. A student stated, the data in figure 9 shows that humans caused mass extinctions. So I know I haven't shown you figure 9 here, but just evaluate the statement. Things you could say, this is a long paragraph, but in support of the statement, there's been a decrease of mammals in some areas. When humans enter areas, the decrease could occur at different times in the area, so it's not suggestive of worldwide climate change. For not supporting this statement, it only shows that large animals... Okay, so this is very specific to the question, so it's not really helpful for you to answer that. But that's the figure's there. So if you wanted to have a go, just have a look at that figure, pause there. But let's move on to the next. So the final question is to define classification. And let's look at that. So classification is the organization of living things into groups which have similarities. So thank you so much for watching. I hope you found this video helpful. There's only one more biology video left to do now, and then I'll be moving on to chemistry and physics. If you found this video helpful, feel free to share it with your friends. Thank you so much for watching. All the best for your exams. Tell me how they go, and see you soon.